Hello and welcome to my presentation. My name is Kedus Kinfe and I'm a rising senior at George Mason University where I'm a biomedical engineering major. And this is my presentation on the activation of FGFR1 protein using a new method in plasma membrane derived vesicles. So first, some background. Receptor tyrosine kinases, or RTKs, are transmembrane receptor proteins, and there are quite a few of them, as you can tell by this illustration here on the right. But this research is focused on fibroblast growth factor receptors, also known as FGF receptors, shown right here. There are a handful of FGF receptors which have similar mechanisms and structure, but we'll just focus on one, FGFR1. FGF receptors regulate cell growth, differentiation, proliferation by transducing signals from the extracellular matrix by signaling proteins known as fibroblast growth factors, FGFs for short. So this magnified illustration right here shows the structure of the FGF receptors with an extracellular domain with three parts, domain 1, 2, and 3, with an acid box in between domain 1 and 2. And the FGFs, the ligands, they bind to between domains 2 and 3. There's a helical transmembrane domain, and there is a tyrosine kinase domain where phosphorylation occurs. So phosphorylation is when pretty much the membrane protein activates. So to do this, RTKs interact laterally along the membrane. So when they come together, they form the structure something along here, where the tyrosine kinase being in the intercellular membrane, and they kind of phosphorylate each other in this case. So yeah, this is usually done in the presence of a ligand where it's much more significant and there's a lot more phosphorylation. When it does activate, it leads to it starts the cascade of cell signaling proteins into the cell, by which pathway causes the cell to activate some sort of response, be it cell growth, differentiation, proliferation, etc. So, because of RTK's significant role in cellular development, their inhibition has been identified as a target for crucial drug therapy of some select cancers, such as kidney cancer, breast cancer, and other growth disorders such as dwarfism. However, new drugs that rely on inhibition of these receptors currently do not live up to their expectations, generally due to a lack of understanding, quantification of the mechanisms of how these RTKs, like FGFR1, function. However, actually studying the interaction between RTKs is pretty difficult due to the heterogeneity of cell membranes under normal conditions. So that's where vesicles come in. Plasma membrane-derived vesicles are these cytoplasm-filled spheres that lack a cytoskeleton, allowing for a homogeneous distribution of membrane proteins. In addition to that, they do not contain any of those internal signaling proteins that cause the cascade down into the cell and could potentially create feedback loops. This all makes it an ideal model system to study membrane protein interactions. So to do this, we actually have to first label the FGF receptor protein with a fluorescent protein through transfection. Then we perform vesiculation. That is actually shown right here with this yellow attachments. The vesiculation process, in short, puts the cell just under the right amount of osmotic stress, forcing it to create vesicles as it undergoes apoptosis, as shown by the image here on the left. From there, we add ligand, ATP, and specific antibodies. The ATP is necessary as a phosphate group is used to phosphorylate the tyrosine kinase domain. The ligand, as discussed before, introduces this phosphorylation, and then we want to measure how much ligand it would actually take for 50% phosphorylation. Therefore, the concentration is the independent variable, as we're going to continually change that between our trials. The antibodies, which are tagged with a different fluorescent protein from FGF receptor, are added to observe whether the FGF is phosphorylated, and as it can only attach to the tyrosine kinase domain when it's activated. So to elaborate on that, so here we have the receptor protein, which is attached fluorescence, and here we have the antibodies with a different fluorescent protein to label it. These antibodies only attach to the tyrosine kinase domain when it is fluorescent, when it's phosphorylated, denoted by the red star here. Okay. All right, so now for some qualitative examples of how this all would look under the microscope. So at the top, we have an example of no phosphorylation, and at the bottom, we have an example of phosphorylation. So actually, let's start at the bottom. So the bottom left image, we are just looking for the receptor. So we're looking for the fluorescent protein that are tagged to the receptors, the ones that are right here. And as we can see, the receptors are pretty evenly distributed, and we can tell the location of the cell membrane. Now, if these receptors are truly phosphorylated, we would expect to see a high concentration of antibody along the membrane based as they are attached to the tyrosine kinase domain, correct? And that is what we see in the antibody image when we look for the antibodies, fluorescent proteins, the ones right here. And there's a high concentration of antibodies along the cell membrane, implying that the tyrosine kinase is indeed phosphorylated. Now, in an example of no phosphorylation, we would still be able to see the receptors in the membrane. But when we look for just the antibodies, there is antibody everywhere, but there is no significant accumulation of their concentration, as there's no phosphorylation. There's no tyrosine kinase for them to bind to. The concentration of ligand between the trials changes by a factor of 10. So in this example particularly, you would measure the intensity of the antibody image, and you would determine the amount of activation at this particular concentration, and then you would change it for a different concentration. You'd be able to construct a graph. This graph would take the shape of an S-curve, with the lower asymptote representing the low ligand concentration and the upper asymptote corresponding to the high ligand concentration and denoting the maximum phosphorylation. And using this graph, you would ultimately be able to determine at what ligand concentration there would be 50% receptor activation. This is still currently being constructed, and hopefully you still are interested in that. That wraps up my presentation. Thank you for listening.